In section 6.8, we talk about the functional anatomy of synapses. So it's estimated there are about 100 trillion synapses in the central nervous system. Remember that this far exceeds the number of cells that are found in the CNS because each individual neuron can participate in multiple synapses. And if we look at these synapses, what we see is that they are of one of two types with many subtypes, depending on the neurotransmitter that's released and the receptors that are present on the postsynaptic side of the synapse. And so we can separate them into these two categories. They can either be excitatory, which of course takes the postsynaptic cell closer to that threshold voltage, which we've defined as a negative 55 millivolts in our case, or something close to that, if the resting membrane potential of the neuron is not negative 70. Or these synapses can be inhibitory, which means that they stabilize the postsynaptic cell and make it less likely that they will reach that magical negative 55 millivolts and fire an action potential. So, furthermore, what we notice is this, that there's a good deal of both convergence and divergence that we can observe in the anatomy of the synapses, especially in the central nervous system. And so it seems incredible to think about, but even thousands of presynaptic cells may converge on a single postsynaptic cell, as we've mentioned previously, and this postsynaptic cell either reaches threshold or it doesn't, but all of those cells, those thousands of presynaptic cells, have a say in whether this threshold voltage is reached. So on the left-hand side of our diagram here, as you follow the pointer on the screen, we see an example of convergence. And so we see four, in this case, presynaptic cells that are converging on one postsynaptic cell. And so all of these four cells have a say in whether this single postsynaptic cell will fire an action potential or not. In contrast, on the right-hand side of this diagram, still following the cursor, we see divergence. In divergence, one cell impacts many postsynaptic cells. This presynaptic cell forms a synapse with these four postsynaptic cells, and through that mechanism, one presynaptic cell can impact many postsynaptic cells. It's important as we think about the arrangements of these neurons to imagine these circuits. A part of a circuit from our text is shown on the right-hand side of our slide. And so we see a presynaptic cell here, which is of course a postsynaptic cell to this particular synapse, connecting to a postsynaptic cell. And so we have this cell, which is presynaptic, to this cell, which is postsynaptic, and then this cell, which is was of course postsynaptic to the previous cell, is presynaptic to the subsequent cell in this particular circuit. Now, this is not a branch circuit, but you could imagine that if you superimpose the ideas of convergence and divergence onto synapses, similar to what we see on the right-hand side of our slide, you could imagine some pretty complicated circuits. And in fact, it's those circuits among the 86 billion neurons that are found in our brain that really compose what we know as the human brain. In the second part of section 6.8, we take a look at the anatomy of synapses. And we begin with this simple idea that we have two types of synapses that we find in the nervous system, both of which you are familiar with at this particular point in time. Number one, we have electrical synapses. And so these are simply gap junctions. These synapses, gap junctions, are of course most commonly known from cardiac muscle, where they connect all the cells of the atria, forming this thing we know as a syncytium, and all the cells of the ventricles, both left and right, forming another syncytium. And that means that all those cells work together, contracting simultaneously. So, briefly, although we do not go back over the structure of a gap junction at this point, reserving that for later in our class, we do remember that a gap junction is formed by a number of proteins referred to as connexin proteins, and those proteins simply form a channel that connects the cytoplasm of two adjacent cells. And this means that a depolarization that results from an action potential in one cell can move without hesitation from cell A to cell B. Gap junctions, therefore, provide very rapid transmission of an action potential from one cell to another. We used to think that these were primarily found in cardiac muscle, but now we understand that they're present in other areas of the nervous system as well. Now, of greater interest to us in this particular chapter, because they're much more common in the nervous system, are chemical synapses. And so chemical synapses are the synapses that we've talked about previously, in which we have a presynaptic cell, which is shown here if you follow the cursor. And here we have the so-called axon terminal. This is the very end of the axon. And if we look at the axon terminal, inside the axon terminal, we find these synaptic vesicles, simply vesicles that are full of a neurotransmitter. Some of these vesicles will be what we refer to as docked, like a boat would be at a dock, D-O-C-K-E-D, docked. And so these that I'm pointing to here are docked and ready to fuse with the presynaptic membrane. If we look at the postsynaptic cell, we have this area referred to as the postsynaptic density, where we have a high concentration of neurotransmitter receptors that are found on this particular cell. The synaptic cleft is the name that's given to this physical space that is present between the presynaptic axon terminal and the postsynaptic density here on the postsynaptic cell. And so it's extremely small. Remember, we didn't know that it existed until Otto Louis came along in the 1930s and demonstrated that it did with his frog heart experiment, as you recall from back in chapter 1. And there's about 10 to 20 nanometers that are present between these two adjacent cells. And so you realize that, of course, what happens here is that the neurotransmitter is released into the cleft, diffuses across the cleft, binds to receptors in the postsynaptic cell, and reinitiates an action potential in the postsynaptic cell. And so this is always how a signal is transmitted from one cell to another within the nervous system if there's not an electrical synapse present at that particular location. Now, neurotransmitters, and we will talk about neurotransmitters in this chapter, vary widely. So acetylcholine obviously is the neurotransmitter that's found in the somatic nervous system, the neurotransmitter that's released, therefore, between neurons and muscle cells. But there are many others that play very interesting roles, especially in the central nervous system. And what we see is that sometimes we have co-transmitters that are released. Co-transmitters bind to separate receptors that are found in the postsynaptic cell and play some sort of supporting role in the process of signal transmission. Now, do keep in mind that by definition, a neurotransmitter moves a very short distance, so it's only moving across the cleft. We do have agents that bind to receptors found in the nervous system, epinephrine being perhaps the first example that comes to mind, that are released and they travel through the blood. But remember, we would have to define those as hormones, of course. So, the epinephrine, norepinephrine is a good example. Norepinephrine, it travels across the cleft and is therefore classified as a neurotransmitter. Epinephrine binds to many of the same receptors, but epinephrine is released from the adrenal medulla. It travels through the blood, binding to receptors all over the body, and therefore it is a hormone. So keep in mind, as you think about neurotransmitters, they're traveling a very short distance. In section 6.9, we look at the mechanism of neurotransmitter released. What we notice is this, that prior to the arrival of an action potential, we, as we mentioned previously, have a number of neurotransmitter vesicles that are docked, D-O-C-K-E-D, like a boat, at the presynaptic side of the cleft adjacent to the plasma membrane. So here we see some such vesicles, if you follow the pointer on the screen here. So this, of course, is our axon terminal. And here we see a number of vesicles that are docked there. Now, there are other vesicles within this so-called active zone, which contains a high concentration of vesicles, where the vesicles are floating around there and are not docked and ready to release their neurotransmitter quite yet. But in an active axon terminal, there'll be a number of vesicles that are docked at any given time and ready to go. These vesicles are docked for this particular reason. Let's go down to the bottom portion of our slide. And what we see is that these vesicles are held in place by these proteins referred to as snare proteins. And so the snare proteins, shown in pink and purple, that I'm pointing to on our slide here, hold the vesicles in place and keep them at a ready state for the point at which an action potential arrives. Now, when an action potential arrives, this is what happens. Let's go back up to the part A of our diagram. So the electrical signal reaches the axon terminal, And it obviously depolarizes the axon terminal. And so not only do we have sodium channels that are present and potassium channels for repolarization in the plasma membrane of the axon terminal, but we also have voltage-gated calcium channels. And so we talked previously about how calcium is oftentimes a signaling mechanism used in physiology. We see it here, and we'll see it in the cardiac system study as well. So at this point, these voltage-gated calcium channels open as a result of the depolarization, and both the electrical and chemical gradient for calcium causes the flow of calcium to rush into the cell. A huge concentration of calcium is maintained typically in the cytosol of cells, and so this allows the influx of calcium when these channels are open. And so the calcium rushes into the cell. The calcium then binds to a second protein. So let's go from part A all the way back down to part B. At this point, calcium binds to synaptotagmin, and when calcium binds to synaptotagmin, which is an additional protein associated with these vesicles shown in green on our diagram, this causes synaptotagmin to interact with the snare proteins and causes the fusion, or perhaps partial fusion, of the vesicle with the presynaptic membrane. This is the presynaptic membrane, of course, and then this is the vesicle. And so you see that once that fusion has been initiated, the concentration gradient allows the neurotransmitter to rush out into the cleft, across the cleft, and bind to receptors that are found, as we see here. These are receptors associated with ion channels on the postsynaptic side of the cleft. Now, this fusion happens in one of two ways, as we have listed on our slide. One possibility is complete fusion, and if this occurs, then that vesicle or the plasma membrane material from the vesicle is later regained via endocytosis in other areas of the axon terminal outside of the active zone. But another possibility is this so-called kiss-and-run fusion, and so this means that there's a slight fusion, exactly like what we see depicted in this lower right-hand side of our slide, what I'm circling here. Some neurotransmitter is released, and then it backs off. So all the neurotransmitter is not released. This is more common in synapses in which the transmission is extremely rapid. Because as you would imagine, you could relatively quickly exhaust the number of docked vesicles that are present on the presynaptic side. So the kiss-and-run fashion would allow you a great deal more time. You could release neurotransmitter in short bursts over a longer period of time.